So thanks to the panel. Thanks to everyone for today. We've got one more to bring it home, but what a journey we've gone on already today. From the beginning of collaboration and innovating how we innovate, and what is adaptive biomedical innovation anyway, and the range of stakeholders that we're trying to bring together, to then hearing some of the actual examples of how it's done, whether it was the iSPY story, whether it was platforms that we've seen in oncology and non-oncology, how the stakeholders actually come together. You might have noticed that we've drifted a bit today into the science side. Remember, Gigi said we need to connect science to the payer, and the payers have been a bit of an afterthought most of the afternoon. Right? Tomorrow we'll fix that a bit more, but I think you'll also hear from Ann Barker, who's going to bring it home for us. That may be one of the learnings she talks about as well, but she has many, many insights. And you heard me say we might actually admit that Arizona can teach us something here in Cambridge, and Ann's going to prove it here for us. So Ann is the co-director of Complex Adaptive Systems and the director of the National Biomarker Development Alliance and a professor at Arizona, and has just a phenomenal history in research and leading large science that has huge impact. So please help me thank and welcome Ann Barker.
Thank you, Mark and Gigi, for inviting me. And so I'm going to use slides because I felt that at this time of day that if only about 10% of your neurons were firing, that you should have something to look at. Because if I just talked, you would be all sleeping. So first of all, this little talk is sort of going to be, I think, platform trials for dummies. If we had to write that book. And we all sort of started out that way when we started designing these trials and actually putting them on the ground. And there are so many things that we've learned and so much that we yet have to learn. And some of it actually has already come out today, but, and you heard quite a bit from Laura on where we are in iSPY 2. And I said to Laura last night, I was in a meeting recently where iSPY 2 was used as an adjective, a verb, and a noun. So I figured we'd arrived because, the industry is sort of talking about iSPY 2 now in all kinds of ways. I want to also point out sort of a couple of things that when I was in the Cancer Institute, I always pointed out these statistics because they're so utterly chilling when you think about it. I mean, our cost of healthcare is about $3.2 trillion. It's $10,000. You're spending $10,000 each on health care. I mean, it's staggering. And 80% of that is in these diseases. And I know we talk about it, but we haven't wrapped our mind around it. And they're all going to increase on the order of 30% and possibly more in the next decade. And we're already seeing it in cancer, because as the baby boomers are moving in to any of that will actually admit to being over the age of 55, we're now seeing these increases. And even though we're getting some advances, it's being offset by numbers of cases. So this is economically destabilizing. And we talk about it, but we're just not yet really thinking about it. And if WHO did this report not that long ago now, but they called cancer the pandemic of this century. I mean, as on a global basis, it's going up, 70%. When I was at the NCI, I talked to almost every country in the Mideast and you name it on their cancer burden. And so this is really serious stuff. And when you think about why these costs are what they are, this is one of the reasons. The clinical trial space is fraught with failure. In oncology, about 70% of our trials fail, which is ridiculous. We're talking about a $2 billion investment for most of these drugs. And in other areas other than oncology, it's not quite that expensive. By the way, Homer Pierce and I made this slide in 1998. It is the most franchised slide ever in the history of the world. I see it everywhere I go, and it's actually been redone like 1,000 times. But it really tells the story. I mean, you start out with so many compounds, and you end up with so little. So this is essentially what Laura talked about earlier, and Andy Grove used to be a friend. He passed away, as you know, a couple of years ago now. He used to come to the NCI every year and beat us up about how things were not going his way or anyone's way in oncology. And he talked about knowledge turns, and he compared oncology to the chip industry. Now, there's some huge differences, and we'd argue about that, but he's right. When a knowledge turn takes 18 to 20 years, and it should take 3 to 5, you've got a problem like you just saw before on that slide. Add to that the fact we all talk about biomarkers. The reason I'm working on biomarkers, when I was at NCI, when I was leaving NCI, I said to my friend Janet Woodcock, what's the biggest problem we have in oncology in terms of precision medicine? She said, biomarkers. We don't have any, we don't know how to find them, we don't know how to identify them, we don't know how to use them. And so I set up this little alliance, and I'll tell you a little bit more about some of that as we go through this talk. But so we're all on the same page. Everybody probably knows what a platform trial is, but I just want to tell you my version of a platform trial so you know what I'm talking about. So these trials focus on a disease, and that sounds obvious, right? But it's quite different when you think about it. And Janet mentioned it this morning. It makes all the difference in terms of how you bring your knowledge and how you bring the drugs, et cetera, and how you organize the trial. The designs can be adaptive. They can be driven by Bayesian statistics, which will make your eyes glaze over, but it's actually the best way to get the best answers in these trials. They're done under a master protocol, and a master protocol is just super. As you heard Mark mentioned this morning, that Janet actually had something to do with the evolution of the master protocol many, many years ago. And the industry uses master protocols all the time. But essentially, it allows you to organize something in a way with a whole group of people and a foundational kind of infrastructure. And if you know how to develop drugs, you can put a lot of drugs under this particular sort of rubric, and it saves huge amounts of time and money. And then adaptive trials, we've talked a little bit about this today, but what you want to do and what we're trying to do in precision medicine is subtype patients so that you can adapt within the trial itself. Laura has a goal of adapting within patients. So, when I looked about this morning at the sort of the headline for this meeting is how do we innovate on innovation, this is the way we can do it. And I think you heard in the last panel, these trials are an absolute haven for innovation. You can do anything in these trials. If you dream it, you can do it. So I'm going to tell you a little bit about what we've learned in 2 different trials that I've been associated with, and you've heard about both of them today. One is iSPY 2, of course, and the other one is GBM Agile, which you heard from Brian Alexander. Is Brian still here? I don't see. Oh, there, he's in the back. My association with these 2 diseases is quite different as well. I became associated with breast cancer because I lost my family, or at least most of my female members, to breast cancer. And I helped to set up the Department of Defense breast cancer program years ago. And it was a disease that we do a lot with if you get this disease very early or if you have stage 1. But if you have stage 3 disease, not so good. Essentially, we have about 250,000 cases of breast cancer a year, about 47,000 women die, and of those women who die, about 15% are these locally advanced disease patients. Those are the focus for iSPY 2. And iSPY 2 is a remarkable effort led by Laura as well as Don Berry and an entire team of people, well over 100 people, working together over 10 years and counting. And it's transformative in so many ways. It's Bayesian driven, the statistics are Bayesian, it's adaptive. Essentially, it's based on subtyping these patients into 8 subtypes and delivering to that particular subtype that drug which is going to be best for that particular patient. That's precision medicine. And it's also probably the first trial where we were told without any doubt by everybody who reviewed us doing this trial is that the industry would never support this. They would never give us the drugs. They would never work together. And of course, they did give us the drugs, and they are working together. And as a matter of fact, they even set up a pre-competitive collaboration to actually share data around the biomarkers. So we've graduated 6 agents out of this trial. 4 have gone on to phase 3 trials, and as I'll show you at the end of the talk, we did it in quite a remarkable way. And credit to the people who did the work. It's just an amazing group of people, stayed together all 10 years, everybody's engaged, and I'll tell you one of the things you learn, it's easy to give credit, and it's priceless. And if you don't do that, people will not participate. GBM, I came to because when I went to the Cancer Institute, I was visited by several people who talked to me about brain tumors and asked me to go look at our portfolio and see how much money we were spending. We spend about $500 million a year on breast cancer. I went to look for GBM, and we were spending virtually nothing, and we were supporting a few investigators. So when I started with Francis Collins, the Cancer Genome Atlas, all the way back in 2004, I said to myself, not to the committee that was actually telling me what I should do, but they told me some other cancer that already had a lot of investment. And but I looked at GBM again, and I called up a few of my colleagues around the country, and I said, do you guys have great samples for GBM? And they all said yes. I said, well, okay, we're going to do GBM, because if we build a really good data set, people will come. And it turns out that's the case. Now we have thousands of investigators working on this disease. And this trial, as Brian explained in his panel discussion, essentially we brought together a few people to think about this disease. What can we do? What do we know? What don't we know? And sort of the known unknowns, the unknown unknowns, you know, that old story. And we started with about 30 people. We ended up with about 130 people working together over 2 years. These are long processes in terms of getting it right. And we started, and we got full approval from FDA for our iSPY 2-like trial, stage 2 screen. We were sitting around having a meeting, putting that to bed, and somebody said, why don't we add a second stage to this and just do a phase 3 trial? Rest is history. That makes this a very, very advanced trial. That will, if you come into the trial, essentially you're coming into a phase 3 trial. Essentially, your drug will move through the trial seamlessly and via algorithm. But what you get is a stage 2, which essentially is equivalent to a phase 3 trial. So my thoughts on what we've learned are going to be in the following areas. Number one, I think it's been an honor to actually really be able to enable these trials coming together. And this has all been done in partnership with the FDA. When I was in the government, it was easy to do, but people like Janet Woodcock and Rick Pazder and those sorts of folks, for the nonprofits especially, will work directly with you. As you heard from the pancreas group today, we've set the stage for this with iSPY 2. I think we've seen it time and time again now that if you've got a good idea, you are going to have to counsel with FDA as you go through these. And so I think we have some successful proof of concepts here, iSPY 2 being the most obvious, but there are others that are coming. But I want to talk a little bit about, this is getting down to the dummies part, and that is for planning, designing, what is the master protocol, what do you have to think about, and operations, you know, what's really important. So let's start with planning. And Mark said something this morning about, you know, administering this stuff and how you actually get it done. And so here's some of my thoughts, and I wish my glasses were much better. It's about the biology. And that means that you have to bring people together who actually know as much biology as we think we know. We don't know nearly as much as we think we know. So people ask me, how do you do this? I say you have to crowdsource the knowledge. And you can't presume that if you have 5 people, you know very much. If you have 130 people, say for GBM Agile or the numbers that we have in iSPY 2, we pretty much cover the waterfront. Essentially, you have to come up with a construct to do that. You have to be able to crowdsource this knowledge. That takes an organization of enormous complexity. We had 10 committees and an oversight committee for the GBM Agile trial. We had something very similar for iSPY 2, and in iSPY 2, all of those committees have continued. You have to look at all the alternatives. This may not be your best shot. Maybe there are other kinds of trials that would be better, but for something like the 2 diseases we're talking about, it turns out to be the right choice. You know, you got to have people think together, and that's really hard sometimes, and you have to enable that happening. You have to engage with the FDA, as I said, early and often. Don't surprise the FDA. That's a very bad idea. Talk with them as you go through the trial. And if it's a Bayesian design, educate the investigators. I can tell you most people do not understand Bayesian statistics, and they will not understand it until you educate them. And so I've seen this too often where they think they understand, but they actually don't. And it's a team. You have to build the trust, and you have to build the confidence. And if you look at what we did, this is the original management structure from iSPY 2, hasn't changed all that much. All those committees are still in place. All the oversight groups are still in place. This is the construct for the planning and design of GBM Agile. That involved 130 people from several countries, and as you can see in there, we have China and the Australians and the Europeans in different colors. But these committees were unbelievably effective, meeting almost weekly for 2 years. The next phase of this, which I'll come to, it kind of depends on some of these big lessons here. Number one, you have to know how you're going to implement these trials when you start. That's a lesson learned on my part a few times. And I think Laura and I think Brian will agree with us. You know, you get caught up in what's fun and cool and exciting, and you forget, starting over, you really have to know how you're going to set it up and how you're going to implement it, and more than that, how you're going to pay for it. And if you haven't done that, then you'll be where Laura and I was out, you know, going to raise money when we were fortunate, but it's hard. So everything has to include everybody. All the stakeholders have to have a piece of this, as you saw up here before. Be clear on your IP policies. Boy, if you are not clear on your IP policies, you will pay at the end of these, because you would be stunned. This is not the private sector. You'd be stunned at the academics who want to own stuff when it's too late to own anything. So you have to, I'm saying that about my friends, but it's true. The other thing is develop the funding strategy early, both to pay for the trial design and also to pay for the trial setup. And as soon as you can get to it, determine how much it's going to cost to enter a drug into this trial. If you don't know that, you can't actually make a very clear presentation to a pharmaceutical company. Identify a number of agents. Make a pipeline early, because you won't get them all. You'll get some, but make sure you put the ones you want on the list, and you put those on the top of the list. And then these are all generally designed and operated through nonprofits. Most nonprofits did not set out to become these clinical trial management enterprises. The diffusion of this has to be to the private sector, or there has to be some new kind of CRO that's going to emerge here. I hope everyone at MIT is listening, because this is a place where we have to do something. Most CROs do not get how to do this. That's why a lot of nonprofits are getting in this business. Design. The team actually must agree on the overall goals to start with. And this is my favorite line from, I think this is from Don Berry, a p-value is not a goal. No one actually cares about p-values anymore. You want to know what you want from the trial. I mean, what are going to be the endpoints that are going to be important beyond, obviously, survival? You're going to use the data as it accumulates, and And that will change your trial parameters if it's an adaptive trial. So plan those adaptations up front. You can't do that after you get into the trial. You can, but that requires all kinds of extra work. You have to define how you're going to graduate drugs and also how you're going to stop for futility. And if you don't have those, as you heard from one of the questions this morning, you know, how do I know when I have a futile drug? As I said before, Bayesian statistics is a great way to run your trials because it offers huge advantages in terms of biomarkers, developing longitudinal models, which we need absolutely for these trials, and actually subsetting patients and the overall flexibility of a continuous learning system. This is the only way to get a real learning system in these trials. However, these designs are extremely complicated. They require simulations that cost lots of money and take lots of time. So you have to really plan for the simulations, and you have to know what you need. The time machine, which you've heard mentioned several times here, has nothing to do with Orson Welles. It actually is just over time. If you have a lot of controls, FDA says that in certain situations you can use all those controls, and it makes a huge difference instead of a concurrent control. And we actually started this in iSPY 2. And make sure that everybody understands the design. There's nothing more embarrassing than getting in front of a group of investors and having one of your PIs not understand the design, which has happened. Okay, this is essentially our framework for iSPY 2. It doesn't look exactly as it did before because there's some things fine-tuned in there over these years, but it's it's driven by intense science, very, very high level of biomarker information in this trial. And as you can see, the unrelenting and ever-present hand of Laura, if you get through this trial and you still have residual cancer burden, you still have another shot. So as you can see here, this trial, actually the one we've been talking about, is quite remarkable in so many ways, and we're about to take this to the next phase. GBM Agile actually follows essentially that design with a couple of exceptions, and what you'll see here is once your drug graduates, it's going to go on to a stage 2, which is a phase 3 trial, and that's a 50-person kind of trial. And everything up to that point is randomized. This is fixed randomization. The protocol, the master protocol, is that's what it's all about. That's what you have to give to the FDA, and then they will start shredding it and telling you what you have to do differently. And you'll have some meetings where, you know, a lot of people will agree with you, most will not, and then you sort of go back and fix things. So everybody's involved in developing this protocol, which takes an enormous amount of patience on the part of everybody, because having that many people edit anything is always a problem. So you're going to have a lot of experts, and they will know how to do this. They don't know how to do this. What they're going to do is try to bring back what they've always done. Don't let that happen, because if it does, you'll be doing what you've always done. You want to have this protocol reviewed extensively by the on-the-ground folks, the clinicians who have to really implement, and the patients who you're going to try to recruit into the trial. And then biomarkers are key to all these protocols. We could talk about that for days, but can they be reproducibly measured, and how are they going to be used in standard of care? Those are two questions you have to ask. Review the protocol as often as you can with FDA. Identify your DSMB up front. They're going to be so important, and you need to know who they are, and they need to know who you are and what your expectations are. If you use a CRO, make sure that they know what the protocol is and what has to be done, and don't put off the easy stuff. Everybody puts off the informed consents, which take enormous amounts of time then, because when you try to do it at the end, or the adverse events, how are you going to do those? And then this protocol is magic because you have an appendix that lists essentially all the drugs that are coming in, but then for each of the drugs, you have a separate appendix. So you don't have to go back to FDA every time you want to add a drug or a combination, and it makes it enormously easier to do. Operations, I'm not going to say much about this. This is where the payoff comes in terms of a learning system. And I'd say iSPY 2 is probably one of the richest learning systems that we have in science today. But whatever your operating plan is, you have to understand this is forever. Okay, whatever you set up, it can't be for the next 4 days or the next 30 days. These trials, these are platforms. They can go on forever. So you have to have that kind of operational stability built in. Data. Measure what you need, not what everyone wants. This is very expensive and very hard. And I think Laura will tell you the same, is that if we measured what everyone wanted, we'd be out of business. IRBs are really different. Laura was ingenious. She synced up the IRBs when we started iSPY 2, but we still have issues, and IRBs are all different. We need central IRBs for these trials. Don't assume how many eligible patients you're going to have that actually go on trial, because having a patient eligible is not the same, and it costs you a lot of money to screen these patients. Plan for change. In breast cancer, the drugs are getting better and better and better. So what does that mean to us in iSPY 2? We have to have only great drugs in iSPY 2. Because if you come to me now and, you know, I want you to go on this trial, and someone says, well, I can go get Pimbro, it's better, you know, why would I want to go on your trial? So every time that we get a new drug approved that's a little better, it has to go into standard of care, and that changes what we can do in these trials. So you have to have, and Bayesian statistics, by the way, allows you to do that. Advance the biomarkers. These trials should be as much about biomarkers as they are about drugs. And if you want to do anything at MIT, join me in the fight to actually get biomarkers approved by the FDA. We approve about 1 per year since 1994. Where am I? Okay, I've lost my site here. Support the team, and as I say, give credit. It's the simplest thing to do, and everyone will take pride and ownership. And it's about reducing knowledge turn, so you need to ask yourself pretty frequently, are you doing that? So has this worked? Yes, it's worked. As I said, this is something that the iSPY 2 team is really proud of. It just got shown down in San Antonio. But 6 drugs graduated and 4 in phase 3 trials. And we started with 3 drugs, and we have, I think, only 1 on there that's really stopped for toxicity issues. We really screen these things very, very carefully. So the iSPY 2 ecosystem is big. It's impressive. It's based on knowledge, and I think it's the kind of ecosystem that will actually allow us to build the learning systems for the future. So these are my takeaways. These trials are potentially transformative. Not yet, because it's not diffusing into the private sector at the rate that we need, and Janet and I've talked a lot about that. Everybody has to get credit at every phase, and if you don't do that, especially in academia, as I said, it's free and it's priceless. So be bold in the design and reach beyond where you think you can go. If it makes sense, discuss it with the docs and then discuss it with the FDA. Build models that can validate biomarkers. The biggest barrier in precision medicine, as I said, is that. We did it with PCR. Pathologic complete response is the first surrogate endpoint that's been approved by the FDA in years. And it was done through this learning system. And now you can get accelerated approval using PCR, which is amazing. Build models, essentially, for biomarkers. Think and act global. You're going to need, actually, immunotherapies are going to soak up all the patients on the planet. So, you know, we have to understand this is the only efficient way to use patients. And so you have to, for example, as Brian said, we got China and Australia and also eventually Europe in our GBM Agile trial. There are 14,000 patients in this country. There are probably at least 40,000 in China. So, and they have a big need there. And then how are we going to open up these learning systems, and how are we going to open up these data? That's a question that we all need to deal with. And then how can we use the platform trials to cut through and change the regulatory perceived barriers and gain these registration for the meritorious agents that we know are out there, and then finally actually convince the industry. So this, I think, is the major barrier, and that is essentially nonprofits are running these things. I don't think that should be their goal in life. The pharmaceutical and biotechnology companies have to embrace these trials, and I think that's going to come mostly from the regulators. So I'm going to stop there and tell you that essentially the future is about change, and I love this slide from Darwin. So it's not the strongest of the species that survive, nor the most intelligent, but the ones most responsive to change. And boy, do we have good news on that front, lots of change. The future, data and artificial intelligence are going to drive these trials, and it's coming so quickly, it's going to make your head spin. Thank you very much.
Thank you, Anna.